Good morning. Today I'm going to discuss on glycine metabolism. Glycine is an aliphatic amino acid. Other members of this category are alanine, valine, isoleucine, and leucine. And glycine is a non-essential amino acid. Today I'll be seeing various biosynthetic pathway for production of specialized products derived from glycine and associated in bond disorders. So, glycine is a non-essential indispensable amino acid. It is optically inactive and glucogenic. So, in subsequent slide, we will see how it is glucogenic. What is the metabolic fate of glycine? So, glycine can be derived from serine, threonine, N5N10 methylene tetrahydrofolate, and glyoxylate. So in this picture, we can see it here. It is converted, serine is converted to glycine in the presence of enzyme serine hydroxymethyl transferase. It requires the tetrahydrofolic acid, which act as a acceptor of this hydroxymethyl entity of this serine. It loads on itself and it converts the serine into glycine. Second is from threonine. So it can also be derived from threonine in the presence of enzyme threonine aldolase. So threonine is converted to glycine and acetaldehyde. So there is a simple breakdown. Splitting reaction is there. One portion, one fragment is converted to glycine and another is to acetaldehyde. Then it is also derived from glycine synthase enzyme. That is also the glycine cleavage enzyme, as the reaction is reversible, can happen in both the direction. So we can see it here. Glycine can be derived from carbon dioxide and ammonia, and the one carbon species being derived from N5N10 methylene tetrahydrofolate. In the presence of PLP, this enzyme glycine synthase converts all these substrate into glycine, and tetrahydrofolate is unloaded now. And the fourth method is right from glyoxylate. So glyoxylate is an keto acid of glycine. So via transamination reaction, so enzyme is glyoxylate oxidase. It converts into the glycine. Now, catabolism of glycine. So glycine is catabolized by following three ways. First is conversion to serine, oxidative cleavage into carbon dioxide and ammonia, oxidative deamination to glyoxylic acid, and it can be decarboxylated to formaldehyde and carbon dioxide, converted to malate, converted to oxalate and excreted. So we can see it here in this particular reaction. Glycine can be converted back to serine, and this reaction is both for synthesis of glycine. Glycine can be synthesized from serine as and vice versa. Like serine, glycine can synthesize serine. So the glycine, the second is like glycine, how it is converted to carbon dioxide and ammonia. So the glycine decarboxylase enzyme is removing the carbon dioxide. Then amino methyl group, from the amino methyl group, the ammonia is released by the enzyme amino methyl transferase. And here there is a role of tetrahydrofolic acid, which takes as an acceptor of which acts as an acceptor of methylene group. So this is how ammonia is lost. Complete loss of glycine entity. And this is the reaction. Glycine is converting to glyoxylic acid through deamination reaction. Glycine oxidase is converting the glycine into glyoxylic acid by removal of ammonia. And this reaction leads to the production of H2O2, which is being scavenged by catalase enzyme. Previously, in previous transamination reaction, and deamination reaction, I spoke on this. How the because H2O2 is a very toxic substance. It is to be removed as soon as it is formed. So there comes the role of catalase enzyme, which breaks this H2O2 into water and oxygen. Further, this glyoxylic acid can be converted into, it is an keto acid of glycine, It can be converted to malate, which can be utilized in the citric acid cycle. It can convert to oxalate, which can be extruded through urine and then converts to formate in carbon dioxide. Formate is an important one carbon species, can be utilized in various metabolic reaction where there is a use of one carbon species in the form of formyl group. It's being loaded onto the N10 carbon of the tetrahydrofolate. Now move on to the metabolic importance of glycine. Glycine is important for synthesis of heme, synthesis of glutathione, that is gamma glutamyl cysteine glycine. It is also utilized for synthesis of purine ring. So this is the purine ring. Purine ring, this carbon 4, 5 and nitrogen 7 are being contributed by glycine skeleton. Synthesis of creatine. Creatine is consists of three important amino acids: glycine, arginine, and methionine. So conjugation of it is also used for synthesis of bile salts, glycocholic acid, and glycosinodeoxycholic acid, which were initially in the unconjugated state were cholic acid and the chinodeoxycholic acid. So its important role is there in detoxification reaction. Say for instance, the glycine can be used to remove the benzoic acid in the system, in the biological system. So it converts, glycine can convert to hippuric acid and can be excreted out to the urine. Then glycine has important role in collagen formation. It's seen at bends or turns in collagen. Every third amino acid is glycine. It is also used for synthesis of glucose. It is constituent of various tissue proteins, hormones, and enzymes. So in this slide, we'll see the conversion of glycine into creatine. So glycine in the presence of arginine, it converted up into the guanidoacetic acid with the loss of ornithine. The enzyme is glycine arginine aminotransferase. This enzyme is present in the kidneys. Further, this guanidoacetic acid is converted up into the creatine. Here, the S-adenosyl methionine act as a donor of methyl group and itself converts up into the S-adenosyl homocysteine. So methyl group is added here, and in this entity converts up into the creatine. And further, creatine can be converted up into creatine phosphate by accepting one phosphate via it is attached to the creatine via the high energy bond. So this creatine phosphate act as an high energy bond containing compound and which act as a storage form of energy both in brain and the kidney and the muscles, especially in the muscles. And further, it can be converted into the creatinine with the loss of phosphate group. It is an spontaneous non-enzymatic reaction. So the enzyme is creatine kinase, which is present in the muscles. This guanidoacetate methyl transferase enzyme is present in the liver. So there are various isoform of this enzyme, creatine kinase, and which is clinically very important. There are so many three forms like BB, and MB isoforms are there. So in muscle dystrophy, this isoform is raised, while in brain it is BB, and while in heart it is CKMB is drastically raised in myocardial infarction and other heart disorders. Then, this is the overview of glycine metabolism. So we can see it. This zone shows the synthetic pathway, how the glycine is derived in the body, in the living being. So from protein degradation, the glycine is there. Then serine can be converted to glycine. Threonine can be converted to glycine. Then this carbon dioxide, ammonia, and one carbon species in the presence of glycine synthase enzyme, they lead to the synthesis of glycine. Further, what is the metabolic fate? Glycine can be utilized to synthesize, can be used to, can be cleaved up into the ammonia. Ammonia can be utilized for synthesis of urea. It can also be converted up into the one carbon unit, which can be utilized in various other metabolic reactions. via serine it can be converted up into the pyruvate, and from pyruvate it can be glucogenic. This is how it is a glucogenic amino acid. Then serine can be converted up into the cysteine. Serine can be converted into choline. So indirectly there is a role of glycine in synthesis of these products. Then glycine's keto acid is glyoxylic acid, which leads to the synthesis of oxalate, which is water soluble and can be easily excreted out of the urine. Then creatine, via synthesis of creatine, it can lead to the synthesis of creatine phosphate, which can be converted up into the creatinine. And creatine phosphate is a high energy bond containing compound which is present in the muscles and brain. Then C4, C5, and 7 units of purine rings are being contributed by the glycine. Then synthesis of ELA. In the first step of heme synthesis, glycine along with succinyl coenzyme A serve as a precursor for heme synthesis. So for synthesis of ELA, alpha amino, delta amino linolenic acid. So this is the synthesis. This is how it is contributing to the synthesis of heme. Then it is utilized in the synthesis of glutathione. So glycine is required for the formation of biologically important peptide, glutathione, that is gamma glutamyl cystenyl glycine. I already discussed about this important peptide. Then it is important for synthesis of bile salts, cholic acid, and um, cholic acid can be converted into the glycocholic acid by simple conjugation reaction. Then it can conjugate with the hippuric acid, with the benzoic acid, leading to the formation of hippuric acid. This is how it is scavenging the benzoic acid from the system. And this is about the disorders associated with the metabolism of this glycine. So we'll see here. The first is the non-ketotic hyperglycemia, and this is the defect in glycine cleavage system. The enzyme is glycine synthase, that is the enzyme of catabolic pathway of glycine. And this disease is characterized by elevated serum level of glycine, leading to the severe mental deficiencies and death in very early childhood. At high level, the glycine acts as an inhibitory neurotransmitter, leading to the neurological defects as well. The second is the glycinuria. The glycinuria is also an inborn error, which is being characterized by increased excretion of glycine through urine, despite plasma concentration of glycine is normal. Since plasma level are normal, so glycinuria occurs probably due to defect in the renal tubular reabsorption of glycine. Then glycinuria is characterized by increased tendency for formation of oxalate crystals. Next is the primary hyperoxaluria. So it is also an inborn error of metabolism which is characterized by high urinary excretion of oxalate. The metabolic defect involves a failure to catabolize glyoxylate, which therefore gets oxidized to oxalate. So this is the spectrum of disorder concerned with the glycine metabolism. Thank you.